So today is the first of two talks in the next two sessions coming from Joseph Powell's lab down under. And thank you to speakers as well as attendees for coordinating the time change so that we could all be live discussing these together. And today, Dr. Drew Neavin, is it Neavin or Nevin?
Neavin.
Neavin, neither. Okay. Dr. Drew
Doesn't
Nevin
look like it.
from Joseph Powell's lab is talking on stem cells for population genomic studies. Their group has put out several studies as well as sort of reviews looking and thinking about how to use many pluripotent lines to understand human diversity, which of course for us in this series is super interesting. So we're very glad to have you speaking today, and take it away. Thank you so much.
Great. Thanks, Carlo. Um, as Carlo said, I'll be talking about population genomic studies and stem cells, and we've developed to help through this process as well. I'm quite happy to keep this informal, so if you guys want to ask questions throughout, if something's not making sense, just feel free to raise your virtual hand and I'll try and keep an eye on it. So today I'd like to start by acknowledging the traditional owners of the land in which this work was conducted at the Garvin Institute, the Gadigal people of the Eora Nation. And I pay my respect to their elders past, present, and emerging. And I'd like to start by talking about stem cell villages, which is a method that we and other people have established over the last few years to help us really scale stem cells to a level that we can actually do population genomic studies with them. And I'll take a little bit of an intermission within the stem cell villages to talk about demultiplexing and a method that we've developed in Muxify to really help with this approach. And I've seen some previous videos from this seminar series where other people have also talked about some of these challenges. So I think this should be quite in line with some of the things that you guys have seen previously. And then I'll also go into some results and lessons from these stem cell villages and future directions and applications that we're using the villages for after identifying how we can establish them and use them effectively. And then I'll also talk about another project that I've been working on over the last couple of years, which is looking at genetic ancestral diversity and specifically looking at it in single cell studies, because this hasn't been very established to date, how diverse our single cell studies are. But since this is a stem cell talk, I'll really focus this in the perspective of stem cell models and the intersection of single cell and stem cell models, how diverse these studies have been. All right, so I'll start with talking about some of the motivations and challenges, especially from our lab and our perspective in applying stem cell villages. And so this really comes from trying to understand the link between genetics and human phenotypes. So we know that there are thousands of genome-wide association studies that have shown us that there are a number of genetic associations that are associated with human phenotypes. But part of the problem is that a lot of these genetic variants are intergenic, which means that they lie outside of genes. And so mapping them to the phenotype and the exact mechanism has been very challenging. And so there's a lot of these steps in between from the genetics to the phenotype. And so trying to understand how that genetics relates through these different this transcriptome and the proteome and the metabolome to actually result in the phenotype that we're looking at. And as I said, more than 90% of these associations are intergenic. And so really linking that genetic variant to the gene that it might be regulating is really important for us to understand these relationships. And so one way that we approach this is by using expression quantitative trait loci, or EQTLs, to provide this functional link between genetic variants that are associated with GWAS and what's happening in a cellular, in a cell. And so here in this pictogram, I'm showing you what a EQTL might look like. So we have a genetic variant where you can either carry the A or the G allele. You can either have two Gs heterozygous, you're either A or G or two A alleles. And here I'm showing that through long distance regulatory mechanisms, the A allele seems to result in a higher expression of this gene, where the G allele has less expression. And so if we assay RNA with RNA sequencing or other single cell phenotypes, what we can do is assay this across hundreds or thousands of individuals and then look at the association between their genetics and the expression. And so individuals who carry two G alleles will have the lowest expression on average as compared to people who have two A alleles who will have the highest expression on average of this gene. And we can fit a linear model that allows us to see this association. And we do this not just at one location across the genome, but across the whole genome, looking at variants that are nearby genes across the whole genome. And there have been a number of initiatives, but I think that one of the most notable over the past 10 years has been the GTEx initiative to look at really the context specificity of EQTLs. And so they've been focusing on looking at regulation of gene expression across different tissues and a lot of different tissues. There are a number of EQTLs that are specific to different tissues, which means that it's really important for us to be studying in the right context to understand genetic regulation. But this study has used bulk RNA sequencing, and with the rise of single cell RNA sequencing and other single cell platforms and technologies over the last 10 years, means that we can get to an even deeper resolution where we can start looking at the specific cell types within these heterogeneous tissues, because we know that within a specific brain region, there's going to be many, many different types of neurons and glia and astrocytes that are making up that composition. And when we look at it as bulk RNA sequencing, it means we're losing the resolution of specific cell types and their genetic regulation. And so the way we can do this is by applying single cell RNA sequencing. So we can either capture with droplet-based methods or place-based methods, and then identify different cell types within these our single cell capture, and then look at EQTLs within each of those cell types. And there's some interesting methods that are coming out that will allow you to do this in a more transitional states instead of just classifying by cell types, but I think Anna will probably talk to that in next month. And so definitely keep attuned to her talk. But typically what we would do is pull out one cell type, so say the brown cell type, and look at the association. You can see here there's no association for that brown cell type, but for the pink cell type, there's a much stronger association, with the blue cell type having intermediary association. And so this is one way that we can think about context specificity for genetic regulation, and trying to understand how genetics regulates different genes expression in different cell types across the human body. And so I'm probably preaching to the choir here, but we find that stem cells are quite a nice model for us to understand genetic regulation in different contexts. And so some of the main advantages, which again, everybody here I think will be very aware of, is that they're self-renewing normal karyotypes. And we can then look at them on many different cell types by differentiating them into these different tissues. And one of the main benefits for us from a population genomics perspective is that we can collect them from many different individuals. And so we can have hundreds or thousands of different stem cell lines collected from different individuals, which allows us to apply these population genetics approaches in stem cell lines. But as everybody here probably knows, there have been major challenges with applying stem cells to population genomics, which include that they're both expensive and time consuming. And each stem cell line is typically cultured in a different cell culture well, which means that we have inherent micro-well effects that cause batch effects, which confound our ability to detect EQTLs or other cellular traits. And so with that, we wanted to try and resolve some of these issues. And so we and others have applied village models. And these models take cell lines from different individuals and pool them together before doing any differentiation. So in one dish, you have maybe 10, maybe 50, maybe 100 different cell lines all pooled together. And then they go through this differentiation trajectory down whichever lineage you're interested in to get to a cell type of interest, but doing that all together. So we resolve some of the batch effects that we're seeing. And it also allows us to to reduce cost because we don't have to culture each of these stem cell lines separately. They all get differentiated together, which really reduces the scale issue, reduces the cost issue, and reduces the batch issue. But of course, as you're noticing, and I think previous talks have also in this seminar series have also talked about this, we need to be able to then at the end point look at each of these cells separately, right? So we need to be able to identify which cells belong to each individual. And so we can do that with single cell RNA sequencing and then using demultiplexing methods to help us parse single cells that are from each individual and remove doublets that would confound our analyses. So now I'm going to take a bit of a sidestep and talk about demuxify, which is a method that we've developed to really help with this method of demultiplexing and removing doublets from the analysis to remove that confounding. And so one of the things that happens when you capture a lot of cells in a reaction for a single cell experiment is that you get quite a few doublets. And so with a 10x capture, this is kind of the scale that you're looking at. So when you're capturing few cells, you'll have very low number of doublets, but as you capture a higher number of cells just due to the microfluidics, you get up to about 16% doublets with 20,000 cells captured on a normal throughput technology. With the higher throughput technologies, this shifts a little bit, but it's still the same concept where we have more doublets than we want. And so we need to be able to remove those as well as identify the cells that belong to each individual. And so what this looks like is if we take cells from different individuals and we process them all together on a single cell platform, we're capturing different cells in different droplets. And so we, number one, want to be able to remove singlets from doublets. So we want to be just analyzing our singlets. We don't really want to be looking at doublets because they could be between two different cell states, and it makes it looks like there's a transitionary state between them, which may or may not be true. And so this helps us be more confident in our conclusions that we can draw from our analyses. And we also want to be able to assign each of the singlet groups to the correct individual. And so we want each droplet to be corresponding to the correct stem cell line that we first put into this experiment. And so there are a few methods to do both of these things. So demultiplexing methods, there have been a number of methods that have come out in this area. Effectively, they identify the genetic variants that are in each of your cells or your droplets and matches those to genetic information that you have for your stem cell lines. And by doing this, it can identify which cell came from each stem cell line and which cells likely contain, or which droplets likely contain doublets as opposed to individual cells. And some complementary methods to this include doublet detection softwares. And these are not for identifying individuals that correlate to single cells, but it does help with doublet removal. And so what they do is simulate through a variety of different methods. They simulate doublets by taking different cells that you have captured in your experiment and adding them together and simulating doublets. And then they compare that cellular profile to all of the cellular profiles in your capture, so that I can identify the cellular profiles that are most similar to these simulated doublets, allowing you to identify and remove those doublets. And you might think, why do we even need doublet detecting software since demultiplexing softwares can also identify doublets? So if we just consider cells from two different individuals, so on the left I have cells that in blue from one stem cell line and then cells in green from another stem cell line, if we can consider even just two different cell types, so this more blobby cell type and spiky cell type from these two individuals, and we look at all of the possible kinds of doublets that we can make, you can see that there's lots of different types of combination, both between the different individuals and between the different cell types. And so demultiplexing methods can identify heterogenic doublets, meaning they're doublets between two different individuals, whereas doublet detecting methods can identify heterotypic doublets, which are doublets between two different cell types. And you can see here that these are quite complementary, where the demultiplexing methods would miss some of the doublets that are not between different individuals, and the doublet detecting methods are unlikely to be able to detect the doublets that are between the same cell types. And so using these two methods together, we hypothesized that we would have better results for our analyses and be able to have a cleaner data set for downstream analyses. And so part of this project, which was published last year, was looking at applying all of these demultiplexing methods and trying to identify the best combination of demultiplexing and doublet detecting methods that we can use to assess our data. And so we used a PBMC data set that we previously published in the lab, the 1K data set, which I think was published in 2022, of just under 1,000 individuals from peripheral blood mononuclear cells. And each of these pools had 12 to 16 donors and were captured as a pool of 20,000 cells. So if you remember, that means we'd expect about 16% doublets for each of our pools. And so we applied each of our demultiplexing and doublet detecting methods to each of these pools to identify how these different methods compare to one another. And then we were able to take the singlets that all of the methods identified and simulate new pools that really establish a gold standard for us so that we know which cells are singlets and which are doublets. And we then applied the methods again to these simulated pools to identify the combination of methods that would perform the best in different experimental scenarios. And so when we first applied these different methods, you can see that not all of these methods perform equivalently. So when we apply them to this data set, we see that on average, we have probably about 20% of a doublet rate across the demultiplexing methods, which are on the left here, and the doublet detection methods, which are on the right. And so you can see that on average, it's about 20% doublets about that have been detected, but there are some methods that definitely identify less doublets and some that I definitely identify more. And when we just consider the majority calling method as the correct assignment for that cell, we can use the MCC as a metric to measure the accuracy of these different tools. And so on average, you can see that the demultiplexing methods seem to perform better at identifying doublets from singlets compared to the doublet detecting methods. But there is some variation within each of these subgroups as well. And so we next wanted to look at how these perform on a data set where we have simulated the data and we know what the singlets, which barcodes correspond to a singlet and a doublet. And I'm not going to go into all the detail of this paper, but I do want to talk a little bit about the differences in the size of pools that can contribute to the selection of doublet detecting and demultiplexing methods. And so in simulating this data, we maxed out the number of doublets that we would expect. We simulated this based on the expected projection for doublets, but we max this out at 20% doublets because we know that after 20% of your capture being doublets, you really don't have a cost benefit of sequencing more, and it'd be more beneficial to sequence these across multiple different reactions as opposed to increasing your cells and increasing the doublets. And so if we just pull out the doublets here, you can see that across these simulated pools, we simulated anywhere between 2 individuals and 128 individuals, understanding that some village cultures can be quite large. And so we want to have the capability to look at multiplex pools of over 100 individuals. And importantly here, you can see that with this pools that have fewer individuals, in the blue here, there are a number of doublets that really would not be detected by demultiplexing methods. And so it's really important to include those demultiplexing methods when we're looking at pools that have fewer individuals in them. But once you get to these really large sizes of individuals, we don't have very much benefit from including the doublet detecting methods. And that's because the likelihood of a doublet being from 2 cells from the same individual is less likely when you have many more individuals. And so it seems like when we get up to these higher numbers, we probably don't need the doublet detecting methods anymore. And I won't go through many of the details of this paper, but I'm just showing you the slide that in the end, we identified that under different experimental conditions, different combinations of softwares were really needed to get the best results. And we've built this into an online tool that allows people to enter in different information about their experiment, including the number of cells captured, the technology you use, whether you have genetically multiplexed data, and then it identifies what we found would be the best combination of methods to apply what the expected doublet rate is and the expected number of doublets for each of these different experimental conditions. And so this is available online for people to use as part of the Demuxify package. And so in the end of this project, which was published last year, we've developed this tool, Demuxify, which houses all of these other demultiplexing doublet detecting methods, and then provides consistent documentation for this online, and provides a lot of different scripts and methods to help with migrating these different methods, demultiplexing doublet detecting methods together, and interpreting the results. And it easily integrates with established single cell methods like ScanPy, Surat, and the Loop browser, and we provide documentation on that as well. All right, so now that we have identified effective ways to demultiplex our data and remove doublets, I'll now talk about our experiment that we use to try and identify if villages can be applied for population genomic studies. And so our main research questions that we wanted to answer with this study was, one, how does culturing multiple iPSC lines in a village influence the unique cellular profiles of each stem cell line? And can we apply multiple iPSC lines? Can multiple iPSC lines be maintained in a village over time, passages and differentiations, without losing a single line through those passages or differentiations? And so we designed an experiment across 3 sites across Australia with some collaborators in Brisbane and Melbourne. And so this experiment was carried out in parallel across 3 different sites to look at the impact of doing these types of experiments in 3 different locations. And part of the reason for doing that is because we know that there are stem cell lines that different groups have across the world, and there might be interest in, say, doing the same experiment, but in 2 different locations with different combinations of stem cell lines, and then combining them afterwards. And so we wanted to know how much variation we would likely have from doing this type of experiment across different sites. And so we carried out this experiment across these 3 different sites using the same batches for cell lines and reagents. So we kept everything as consistent as possible, except the location and the people handling the cells. And we took 3 different stem cell lines and pooled them at day 0, which we're calling our uniculture, and captured for single-cell RNA sequencing at that time point. And then we cultured them in this village for 4 days and captured them again at 4 days across the 3 sites. In Sydney, we also cryopreserved some of this baseline village and then thawed and passaged this to see the impacts of cryopreservation on the village and the distribution of cells and their transcriptional profiles. And so in doing this, we're hoping that we can really parse out the impacts that the village has on the transcriptional profiles and the proportion of lines in villages. And so today I'm just going to focus on the fresh results. The results from the cryopreserved samples were effectively the same, and just due to time, I'm going to focus on the results from the fresh samples across the 3 sites. And so we applied Demuxify to our data from the experiment I just described on the last slide, and wanted to look at first the proportion of cells of these 3 different cell lines at the 3 different sites at each time point. So the first thing that we noticed, which we were quite happy about, is we have pretty equivalent proportions of the 3 cell lines at the uniculture. So at the time that we're pooling them together, we would expect this. Sorry, you'll have to bear with me. I'm recovering from a cold, so I'm just doing my best here. So the combination, we have a combination of these 3 lines at uniculture that are really equivalent, but after even just 4 days in culture as a village, we see that we have a decrease in 2 of the lines and an expansion of the other 2 lines. So we see that the yellow line really increases across the 3 sites. Sorry, just give me 1 second.
Take your time. Take your time.
All right, I think we'll be good for a bit now. Okay, so what I was saying is that we're seeing this expansion of the yellow line across our sites. We don't see this expansion as much at site 3. This is partly because this was our site, the site in Sydney. We plated the cells at too low of a density, so we didn't see that the cells really recovered and grew enough for this time point, but because we wanted to do this experiment in parallel, we processed the cells anyway, and so that's why we don't think that we see the same kind of characteristics at site 3. But I'll show you later on that we continually see this kind of outgrowth of some cell lines. And we hypothesized that this was likely because of different growth rates of the cell lines. And I think that you've likely heard this from other groups as well. I know the other groups have presented this kind of data as well. And we indeed tested the growth rates of these different cell lines. And we can see that the yellow cell line has about 2 times the growth rate of these other 2 cell lines, which makes is very concordant with what we're seeing here, that we have this expansion of the yellow cell line compared to the purple and the blue cell lines.
Don't worry if you
And
need
so
to
next,
take a break.
yeah, I'm so sorry. I think it's just a little tickle, so once it goes away, it should be good. It looks like we have a question.
I'll ask a
Um,
question to give you a little bit of a break, maybe.
Thank you.
So previously, we've heard a couple of talks in this seminar series who talked about similar experiments where, you know, the authors were mixing multiple cell lines. And there, what we saw was that they didn't mix in the beginning of the differentiation. They were mixing like, you know, 1 week in. And what we were told is that when you mix them 1 week in, you get more uniform number of cells at the end, towards the end of the differentiation. What's your opinion and experience on that regard?
Yeah, so I'll talk a little bit about that. And I think it really depends on the differentiation. So we've had some conversations with other groups who are working in this area as well, who also do a very similar approach to that, where they wait until the cells are post-mitotic before they pool them together. We think that by accounting for growth rates, we can get around that a bit. And so we can identify groups of cells that will perform well together. And I'll show some preliminary results for that later on. But I do think it really depends on the differentiation that you're approaching. And I think some of the previous talks have talked about neural differentiations where there's quite a long time between pooling them and then becoming post-mitotic and really getting past that neural progenitor state. And so I think that's a scenario in which you could design it a few different ways where maybe you're pooling them when they're NPCs so that when you're pushing them towards a neural post-mitotic state, you would be able to get that more of a good distribution of cells. But I will talk about that a little bit more later. Thank you very much for giving me a little bit of a break there. We'll see how I go for the rest. Yeah, sorry about that. Okay, so we see that village samples are highly correlated. And so the way that we did this is we had run this experiment across the 3 sites in triplicates at each site. And so we looked at the correlation based on these different variables that we've measured, which are replicate, village, line, and site. So between the correlation between our replicates and the correlations on the x-axis here. The correlation between our replicates is really the highest correlation, which we would probably expect, followed by our village correlations, and then by the correlation between samples from the same line, and then across sites. And so what we can see is that the village correlations are really quite strong, indicating that we are probably not influencing the transcriptional profiles of these iPSC lines when co-culturing them. But to look at this a little bit further, we're applied linear mixed models where we can look at the variance that's actually, and so the way we can do this is we take each different gene and fit a model to this gene, looking at these different covariates such as the stem cell line, the replicate, and the village status. And in doing this, we can identify the variance that's explained by these different variables. And so this is on the bottom are figures of what this might look like. So we have the stem cell line on the x-axis, gene expression on the y-axis, and here you can see about 50% of the variation in the expression of this gene seems to be attributed to the stem cell line. So we're seeing quite a large difference between our 3 stem cell lines here, but very little difference between our replicates and our uniculture versus our village. And so they make up a very small percentage of the variance of this gene. We can also add in interaction effects, and these are effects where the combination of 2 independent variables, so our stem cell line and our village, are acting in more than an additive fashion, meaning that they're acting in a stronger fashion than you would see if you looked at each of them separately. And so you can see that here by the slopes of these 2 different lines. So in uniculture, we have a less steep slope. And then in the village culture, we see that we have a more steep slope, indicating that the village by line effect here is much stronger than the uniculture by line effect. And so we were hoping to not see many of these effects, largely because it could signify that we are actually, the village is actually impacting our ability to detect line effects. And so we went through and we measured this for all of the genes that we captured with the single cell experiment. And looked at the variance that's explained by these different covariates. So we'll first start by looking at the variance that's explained by the four covariates on their own. So the replicate, the sites, so the 3 different locations across Australia, whether it's a village or uniculture, and the 3 different stem cell lines. And so across the x-axis, you're looking at the percent variance explained, and then this is a histogram with the bars underneath indicate the an occurrence of a gene. So you can see that the lines explain up to about 80% of the variance of some genes, and there's one gene that's about 80% of their variance explained, with other genes kind of in between and lower than that. The village explains a much smaller proportion of that variance, with the site showing somewhere in between the village and the line effect variation for a number of genes. And then replicate has a very small impact on the variance that's explained, which I think we would expect. And effectively is our lower bound for understanding technical noise here. So this really identifies kind of what we might expect to see for technical noise between replicates. And so then when we look at the interaction effects, I want to just focus your attention first on this orange histogram. You can see that our line by village effects are really on par with our replicate effects, indicating that an interaction between the line and the village is not impacting our ability to detect a line effect. And so this was really reassuring to us that villages could potentially then be used for us to study population genetics without it having a large impact on our ability to detect these kind of EQTL effects. I will also point out that the village by site effect is relatively strong. So it seems that the village has a different impact at different sites, indicating that if you were to carry out these kinds of studies, large scale studies across multiple sites and integrate them, you'd have to be very careful about how you go about doing that, and probably want to have some matching lines between sites so that you can normalize between those 2 sites. And of course, there's a large proportion of the variance that we can't explain with these variables, which would be expected for a number of these genes. And so one of the first things we did was look at the proportion of variance that we could explain for Y chromosome genes. And the reason we did this is it's effectively a positive control. We have 1 and then 2 that were derived from female donors. And so we would expect the line to be the larger proportion of the variance that's explained for Y chromosome genes. And that is indeed what we see here. So this purple color that you can see, it's a line effect, and we can see that that is explaining the larger proportion of variance that we can explain with the variables that we have in hand. And we see very little of either the lighter purple, which is the village color, or the orange color, which is the line by village effect, indicating that they have a very small impact on the Y chromosome genes. So this is, again, just a positive control for us. to make sure that we can identify variables that are regulating gene expression or contributing to variants in gene expression. So then we looked at something a little bit more interesting, which is the pluripotency genes. And we see there's a lot more variation here, which we would expect between the different covariates that are impacting the variation that we're seeing in expression of these pluripotency genes. But most of them seem to be driven not by the village or by the line by village effect. Those would be that light purple and that orange color, but by other covariates such as the line and the site. And so these are potentially what we'd expect. Different people handling cells might have slightly different results. And so again, we were quite happy to see that the village and the line by village were not impacting our ability to look at variation in gene expression. And so next, EQTLs. So these are EQTLs that were identified by the DeBover et al. Paper in Stem Cells in Kelly Fraser's group. And we wanted to see if we could replicate them in our much smaller study. And so we took those EQTLs and we tried to, we looked for effect sizes in our cell lines. And reassuringly, we were able to replicate a large proportion of the EQTLs that they identified. So as you can see here, most of the EQTLs are falling in the upper right-hand and lower left-hand quadrant, with fewer EQTLs falling in the off diagonal quadrants. And this is just in the uniculture. So when we look at the village culture, we can see almost the exact same pattern where we're seeing replication of the EQTLs that had been previously reported. And we next looked at the correlation of these effect sizes. So the effect size of the villages on the y-axis with the uniculture effect size on the x-axis for all of these EQTLs we tested, and we could see a strong correlation between them. And so this indicates to us that probably our inability to replicate some of these EQTLs is likely due to our just loss in power from having so few cell lines as opposed to the village effect. And so we're seeing that the village is performing very similarly to the uniculture lines. And when we pull out a single example here of CHCHD2, you're looking at normalized expression on the y-axis and then the genotypes on the x-axis across our 3 sites. For our uniculture on the left and the village on the right, you can see that they all have very consistent effects. So a negative slope for all of these different associations. And we do notice that the association is slightly stronger for the our village samples, which might suggest that we're actually removing some of the micro-well effects that come into play when we're culturing the lines separately. And so it's able to remove that noise and allows us to identify stronger associations and probably more associations with weaker effects as well. So this is very reassuring that we can probably apply this kind of method for population genomic studies. And so the next thing that we looked at was if we could see dynamic effects across our cells that we captured for this experiment. And so we identified a pseudotime for all of our cells using RNA velocity. And this pseudotime actually reflected pluripotency state. So you can see that the highest expression of the pluripotency state is kind of in the main body of this UMAP. I think of this as an elephant. So in the main body of the elephant, you could see highest expression of PU5F1, a pluripotency marker, and lower expression in the trunk to the upper left. But in that trunk to the upper left, we do see ectodermal and other markers. And this is small proportion of the cells, but it seems that these are some of the pseudotime is really reflecting pluripotency state with the lower pseudotime values corresponding to more pluripotent cells and the higher pseudotime values corresponding to less pluripotent cells. And so similar to how we looked at the variants that could be explained by different covariates in our model, we also wanted to see if how much of the variance could be explained by this pseudotime. And so we can fit pseudotime as effectively a continuous variable here. So we can see how the variation in gene expression is changing across the pseudotime and look at its interaction with cell lines. So you can see in this toy example here, across pseudotime, we're seeing a continual decrease in expression of this gene. And at the higher values of pseudotime, we see much more similar expression across our 3 cell lines, the blue, purple, and yellow cell line, and we see much more variation at the lower pseudotime values. So just to show you an example of what we found here, we found a number of dynamic effects. And here I'm showing you CHCHD2 again, just that example gene that I showed for the EQTLs as well. And we see a pretty strong pseudotime effect for this gene that interacts with the cell line effect. So we see that in our more pluripotent state at the lower pseudotime values, we have much larger variation in gene expression across our 3 cell lines, with this variation being much decreased at the higher pseudotime values, which are our spontaneously differentiating cells. And so we now know that we can use these types of experiments, village models, for looking at EQTLs, but also for looking at dynamic effects across time, which was also very reassuring and interesting. So this will get a little bit to the question that we had earlier. So one thing that we wanted to do towards the end of this paper is look at more cells in a village, because we'd only been looking at, so far, a village of 3 cell lines. And so this work was carried out by Nona Farbecky, who created a village of 18 iPSC lines and then differentiated them into cardiomyocytes and captured them at various days across that differentiation. And when we looked at the proportion of lines across this differentiation, we can see that these cell lines were not selected based on their growth rates. And we do see, again, that we lose some of the cell lines across this differentiation. It's a 15-day differentiation, but we do maintain most of the lines. So we still have 12 cell lines that we can detect at the final time point, although we do see expansion of some of these lines that really takes up the ability to detect some of the other lines. As part of this experiment, we also cryopreserved some of this village at the time of creating it, and then thawed it for multiple passages to see how this large village would perform across multiple passages. And again, we see that while we can detect many of the lines after 8 passages, we do lose a lot of lines in the process. So this was just another indicator to us that growth rates are an important factor here. And this has been shown, as has been pointed out by a few different labs at this point as well. And so this is something that we've been thinking about how we can approach this. And in the interest of time, I'm just going to skip that next slide. So because drawing the same conclusions. So I hope that I've been able to convince you that village culturing has limited impact reliably to look at genetic effects and other line-specific effects. And that growth rates are a really important consideration for village generation and something that we've been thinking a lot about. And that dynamic effects are also detectable with village models. And so now I'm going to talk a little bit about the future directions of STEM, how we're thinking about applying stem cell villages. So this was really picked up by a graduate student, a really talented graduate student who joined the group almost a year and a half ago now, Trevor Atkinson, who, when he first joined the group, agreed to assess the growth rates on all of our cell lines that we had in-house, which is about 200 iPSC lines. And so this was really a feat, and he was able to do this for all of these cell lines, which I have lined up here across the x-axis. And you can see that somewhat reassuringly, these follow a normal distribution, as you would probably expect. So most of our cell lines have very similar growth rates between about 1, and this metric is just a metric for comparing the lines. It doesn't really reflect replication rate or anything, but between a growth rate of 1 and 1.5 is where most of our cell lines lie, with a few cell lines on either tail that have lower growth rates and higher growth rates. And so this means that we can now, in a more educated way, create villages that have more similar growth rates. So we avoid that kind of overgrowing of a single cell line that would prevent us from being able to detect all of the cell lines that we've included in the village. And so one thing that we've done is test this out. So we took 20 cell lines that we had measured the growth rates for, pooled them, and then collected them every day for a cardiac differentiation. And these are the growth rates pulled out of that distribution. And at the time, we didn't have all the growth rates completed yet, so they were a little bit closer together when we first selected them. But I will remind you that in the middle here, this variation is very small between the different lines and their growth rates. And so when we looked at the proportion of different cells that we have across this differentiation at each time point, we select by growth rates. This allows us to be able to detect all of the cell lines that we want. We do, of course, see a little bit of outgrowth of one of the cell lines here, but it's not as drastic as we were seeing before. And so this really allows us to kind of take this to the next step and avoid those overtaking of the village. And because we're a bit at time here, I'm actually going to skip my work on genetic ancestry and probably should go towards the end. But what we're really excited is that these villages can be applied for looking at genetic regulation under different contexts, which is often typical to study outside of using stem cells. And so we can look at genetic regulation across differentiations or under specific stressors or in drug response, and also use them for drug screening and preclinical trial testing as a more representative population sample than just using one or a few stem cell lines. And I am going to skip to the end here just because I think my coughing attack lost me a little bit of time. But I'd like to thank everybody who's been involved in these projects. So Demuxify was done in collaboration with the Single Cell EQTL Gen Consortium. This first QR code will take you to the documentation for Demuxify, and the second QR code is to the publication, which was published last year. And then everybody who helped on the villages. So this was done in collaboration between 3 labs across Australia, Nathan Pelopin's lab at the Institute of Molecular Biosciences at the University of Queensland in Brisbane, and then Joseph Powell's at the University of Melbourne in Melbourne. And then the cardiac differentiation results that I showed as part of the publication were done by Nona Farbecki and Angela Steinman. And then Trevor Atkinson has recently joined and really taken up the kind of next stage of villages in our lab. And Yasmin Bermudez helped with the multiple passage large village effect. And even though I didn't have time to talk about it today, we've been working on genetic ancestry methods for single cell studies and how we can use those to understand the diversity of different individuals that are being included in different studies. And so I want to thank everybody there who's helped with that work as well. And everybody from Joseph Powell's group. This photo is from our most recent lab retreat. So everybody's been very helpful. It's really fun working with everyone. And I'll thank my funding bodies as well. Thanks for listening. Happy to take any questions or just open it up for discussion.
Thank you.
Can I ask one more question?
Absolutely.
When you are mixing those 20 or many cell lines at the end, have you looked under the microscope, and are they actually growing mixed, or do they form separate colonies? Have you looked at them?
Yeah. So I think that would be a bit hard for us to do with our current setup, just because we don't have markers for each of the lines. So we don't have like a fluorescent tag or anything for us to know which line is which when we're looking under the microscope. But we do know that when we sorry, I've lost my train of thought. Yeah, sorry. When we plate a cardiomyocyte differentiation, effectively what happens is we have to plate them at a really high density so that the differentiation goes well, especially in 2D. And so for this differentiation, they were plated at a really high density so that we'd get to about 90% confluency before starting the differentiation. So I think it would be a little bit hard for them to create these different colonies. But I would expect that they won't be creating different colonies, but we should have some results that will hopefully help us answer that in the coming months.
Okay, thank you.
Oh,
Yes. I have two questions. The first one is to measure the growth rates. You use, I think, in the paper RATRAC, right? And you compute the growth rate per day at different. How do you do that? Because I know the RATRAC was designed for microbial growth rate. I don't know if this is also used for works well for cell lines, or what's your opinion on that?
yeah, I mean, so effectively how we did it was we just dissociated the cells at multiple different days. So when we plated them, and then we would dissociate them every day for about a week, and we would count the cells. So we plated different wells and counted the cells across that time frame. And I mean, the dynamics should be the same, right? For microbial or non-microbial cell splitting. And so we just fit a model to that to see what the growth rate is across that time frame. The more recent, that's quite a time-intensive method. And so one of the big things that Trevor really added to this project is finding a method that would allow us to look at growth rates in a little bit of a more high throughput method. We lose a little bit of accuracy there, but it allows us to actually measure the growth rates in a reasonable way. So we have balanced these two kind of needs in the lab.
That's fantastic. And my second question was, like, we have done an experiment which is similar to what you have done, but we have added an extra lineage with the same pool. So essentially we have differentiated the same pool towards an endocrine lineage to a neuron lineage, and just passaging the same pool. And that's probably unsurprisingly, we have found different outliers in different pools. So we were wondering whether growth rates, what you have found that explains the different proportions of cell lines, can be just explaining certain lineages, but not others, which is my fear here. And I don't know how we can study that. I don't know if you have any point of view on that, or you have seen it, or.
Yeah,
before.
no, that's really interesting. We haven't done villages in a lot of different lineages yet. We've done a few of them, but I don't think we've done kind of that controlled village kind of design that you've done. But I wouldn't be surprised. So I think that part of what explains that this variation is growth rates, right? But I think it's also part of it is often in these differentiations in those early time frames, there's a lot of cell death, and some cell lines may be more sensitive to these changes in their environment based on the molecules that are being added. And I think that will be different for different differentiations. And so I think that we can try and control part of that variation by controlling for the growth rates. But the other part of that variation, we may not be able to control as much unless you do an individual differentiation for all of them and take a look at those growth rates. But I think, as was pointed out earlier, there are other approaches for especially some types of differentiations. I think especially neural differentiations, where you could get cells to like an NPC stage, freeze down all of your lines as NPCs, and then pull them at that stage for further differentiation as villages. So I think there are some approaches to get around this, but I think also from what Anna's going to talk about in a few weeks, they did a neural differentiation where they pooled at baseline and were able to get many lines out. And since we're typically talking about hundreds of lines, if we lose a few of the lines in that differentiation, we're kind of expecting to lose a few. And for our purposes, we don't need to maintain all of them. We're just trying to do our best to maintain as many as we can to increase our power. So I think it really depends on kind of what your experimental design is, right?
Yeah, fantastic. Thanks.
Yeah.
Soraya and Timothy, did you both have questions?
Yeah, I can go. I was just wondering if you, well, first of all, great talk, and I really enjoyed the Villages paper that came out a couple of years ago. I was wondering if you looked at if any of the QTLs could explain the differential growth rate across the lines. I think there's a lot of interest in identifying backgrounds that are give rise to better pluripotency and self-renewal.
Yeah, that's a great question. I don't think we were powered to do that in the first village paper, because we only had 3 lines. But we should be powered to do that now with our growth rates. maybe not looking at EQTLs, but looking at associations of genetics with our growth rates that we've measured. And then could do some additional studies with differentiations, similar to what Paul had suggested, where if we see an outgrowth of a line for a specific differentiation, if there's a genetic component to that. So we are thinking along those lines. We just haven't gotten to that yet. Thanks. Great question.
So I have a question that might be, I could invite musings from anyone here because I think I don't think any of us have done it. But if we think, and you mentioned this, Drew, just a second ago, that the experimental design, what you want to look at, controls a lot of this. But in a system where we might not be looking to push every cell to the exact same place, but perhaps see where 10 or 20 or 100 lines might go in a landscape, I think things become quite much harder. You could imagine, 100 lines that in a dish alone might have very distinct behaviors. But if you are one of 100 lines running around when a decision is made, your natural propensity to do left or right or anterior or posterior might be harder to see. I don't know if most of the metrics you were checking along the way was to make sure you had enough cells of each line in the cell state so you could measure EQTLs in a neural precursor versus a cardiac precursor. But if we want to ask, say, I guess it's a much more difficult question to ask about how different lines make different decisions, how could we use what you know from what you've done already to design experiments that are a little more open as far as the phenotypes we want to explore? I don't know if that's too open-ended of a question, but
I'll answer first and then kind of open it up, because I think this is an interesting discussion point, right? I think what Anna will present in a couple weeks in her experiments, they were able to detect something similar to what I think you're talking about, which is kind of like the stochasticity of a line and the propensity for it to go one way or another, and how stochastic that is, right? Is it built into the genetics or the inherent properties of that cell line, or is it slightly variable, right? Am I understanding correctly?
yes.
So one of the things that Anna had done in their paper was look at the lines that didn't differentiate as well. So they didn't get as far down the differentiation lineage, and then they just replicated that differentiation. And this was for dopaminergic neurons. They replicated that differentiation by just doing it for that single line, and they see that they can't get it to differentiate as well. And so it seems like it is specific to that line for on average for that line, it just can't differentiate as well. But I don't think that they looked at maybe the more resolution. And you should ask her when she presents in a month, because I think she'll have
Cool.
a bit more to say about that in the analyses that she did on that front.
Wonderful.
Yeah, maybe linked to this same question is like, how much big can you go in pools? Because I guess there is a minimum number of cells you can get per line to have a good estimates of cell type proportions and representative phenotype, right? I don't know if you have made any power calculations on that, or you are not interested in like the rare cell types, or.
Yeah, I think part of that is just how many cells you capture, right? So you could have a village of 100 lines, and Kevin Egan's lab did something with this, but they weren't looking at single cell outputs. So they were just looking at whole genome sequencing to look at the proportion of lines. So I guess you just have to capture more cells to look at, have enough cells from each individual for each cell type. But yeah, I think I think it really depends on the differentiation that you're looking at and the cell types that you're looking at, right? If you're interested in a really rare cell type, maybe there are better ways to get it than just assaying everything, or you'll just need to sequence more cells potentially.
I go back, hi, very nice talk. I go back on the growth rate quantification that you perform, how reliable is, like, for example, if you tow again the lines, like the same lines, passage multiple times, they maintain the same growth rate, or you think it's some parameters that we have to always keep track before starting with a village approach? And another question is that my second question was about the variance among sites, because you mentioned that you control for media, the same lines, and imagine you share the same protocols. So it's possible that what's the variables there is the personnel that counts, dissociate, and plates. So yeah, if you can comment on this. Thank you.
Yeah, thanks. So remind me your first question.
The growth rate, how
Yeah.
how often you have to check on it, is pretty stable.
Yeah, we haven't actually checked that, but we have some upcoming experiments that I think will reveal that to us. But I think also that experiment where we chose lines based on their growth rates showed that they are probably pretty stable, because we had measured them and then done the experiment at a later date. So we hadn't measured them and done the experiment in the same time frame. I think this also harkens back to a lot of what, like Paul was saying, is that there are multiple variables impacting the proportion of lines in a village. And so this is one way we can try and address it, but it's not a perfect way, and we won't achieve a perfect answer from this, right? And your second question, I'm so sorry.
The village and site variance that was higher compared to,
Yeah.
right?
Yeah, so I think that it has to do with the individuals processing the cells or that location, right? So I think there's a lot of variables that we can't measure in an experiment, which are quite hard. So it would have been very interesting. This is nearly impossible, but it would have been very interesting to have the same individual processing cells at multiple different sites, right? Or multiple individuals processing cells at the same site. And there you can kind of parse out, is the actual location impacting it? Is there something in that environment that's slightly different, maybe different incubators? I'm not sure. Or is it the actual people handling the lines? So I think there are other variables that we can't measure. I can't say exactly what those are, but they seem to be influencing it a little bit.
Mm-hmm. Yeah, exactly. Basically, your elephant UMAP show exactly, basically each lab, how we handle the IPS. Of course, if you maintain them at the really super happy, you clean a lot, you will not have those ependymal or whatever, what was a neuroectoderm. Maybe other labs, they don't care if there is a bit of differentiation, how you passage them. Anyway, but thank you. Yeah, I read your paper. We present a lab meeting some time ago, so it's quite interesting.
Lovely. Thanks, Sarah.
Any other questions?
We will apply the village and keep you posted.
Sounds good. It's always nice to learn from what other people are doing in the field as well. So it's always good to hear what other people are thinking about and doing.
Mm-hmm. And we are applying your system to the multiplex. So, yeah.
Feel free to reach out if anything is challenging with that.
Okay. I will refer to to my colleague.
Sounds good.
Well, thanks so much, Drew. And Pao, did you have a comment or question?
Yeah, I was about to say something. We were, one of the hypotheses we wanted to test in the lab is like how passages affect the lines, and indeed whether the growth rate is changing because of those passages. That's something that probably should be connected also with kind of sequencing, but I think nobody does that. Of course, the cost is huge. I don't know if you ever had any access to this kind of data or thought about this, or what's your opinion?
I think that's also similar to Soraya's question, right? We definitely haven't done that experiment yet. I think our personal experience is that of course, if you passage the line multiple times, you will see different characteristics, which likely come from, say, stereotypic abnormalities that are being picked up or something else is being picked up in culture. So we do try and batch ourselves so that we have a large number of files available to us that have a lot of QC on them. And then we do that relatively frequently. But I think that's a great experiment to be run. And I'll be really interested to see what you find about it, because it will have large implications for, I think, a lot of people in this area.
Thanks again, Drew, and looking forward to another talk from your group, Joseph, next month. Really interested in these methods and thoughts. They're really going to affect how we understand differences between humans in the next decade or so. So thanks a lot, and see you next month.
Thanks. Bye.
Thanks, everyone.